Hello everyone, welcome to my talk. I'm Melania and I'm a PhD student at the International Max Planck Research School and Free University in Berlin. Today I would like to give you an overview on the design of so-called distributed cell classifier circuits. I would like to start by introducing cell classifiers to you. Cell classifiers are very little synthetic biological circuits or biodevices that may be programmed to classify a cell state in vivo, so while being inside of the cell. They may be programmed to distinguish, for example, between healthy and cancerous cells based on their microRNA differential expression profiles. And today I'm going to focus on such classifiers. Here we have a little example of how they work. Let's imagine that this is a healthy cell and this is a diseased one. Those little yellow circles are cell classifiers that are introduced into the cells using, for example, viral vectors. When inside, the circuits may spot the molecular differences between those two cells and based on that classify the cell condition as healthy or diseased. Then, in the latter case, the circuit may trigger a proper response, which may be, for example, releasing a drug causing cell apoptosis. The long-term goal is to create a new kind of personalized treatment for cancer patients. One of the reasons why cancer is still so unbeatable is the cancer cell heterogeneity not only between different patients, but also between different cells belonging to the same tumor. So the goal is to create a single treatment for a single patient. And here, the samples may be collected from different sites of the tumor, as well as from the different sites of the surrounding healthy tissue. And based on the acquired data, personalized circuit design may be prepared, and the treatment may be delivered back to the patient. To design cell classifiers, we work with microRNA expression profiles that may come from different platforms. But usually they consist of unique sample IDs that are followed by their annotation, which basically says which sample comes from healthy and which one from the diseased tissue, and the annotation is followed by the microRNA expression profiles. After quality control, normalization, and other data preprocessing procedures, to obtain a clear-cut information about the microRNA expression, we discretize the data according to a certain threshold into high and low microRNA levels. Then we employ our binarized data to look for relationships between the microRNA profiles and the sample annotation, and such relationships may be represented, for example, by logic functions, like this one here in the middle. This function says that microRNA A or microRNA B has to be upregulated and microRNA C has to be downregulated to classify a cell as cancerous. The logic functions that are the most accurate ones and are also at the same time the least complex ones are baselines for the circuit assembly in the wet lab. So to sum it all up, what we are searching for are logic functions that classify the samples with the highest possible accuracy, but are also at the same time fairly simple and satisfy all additional wet lab constraints, as not every single function may be translated to a wet lab design. First classifier architecture we worked on were single circuit classifiers that are basically large single functions consisting of up to 10 microRNAs. For this, we created a method that allows to find globally optimal functions using logic solvers, but large single functions may not comprehend the cell diversity very well, and therefore the single circuit architecture is potentially less robust to the cell heterogeneity. Also, as you may imagine, more complex functions pose difficulties for the wet lab assembly. Therefore, we introduced a new theoretical architecture called the distributed classifier that consists of a few different, very simple functions that classify cells according to a predefined threshold function. Here, a set of very simple functions is designed to decide collectively on whether the cell is cancerous or not. It can be compared to voting. Each function votes for a particular cell, and then the votes are summed up, and according to a predefined threshold function, the drug is either released or not in those cells. Our goal was to test whether this theoretical architecture may be more robust to the heterogeneity and noise in the data. However, we needed to introduce heuristic approaches to compute the classifiers due to a bit more complex search space and new optimality criteria we introduced. As the design of classifiers begins with data binarization, we also tackled the problem of binarization-related information loss. The transition from the continuous to binary world usually results in some data resolution loss, so we looked for binarization methods that will allow to somehow retrieve this lost information, and at the same time will help us to find more robust microRNA candidates. For this, we chose the method proposed by Wang et al., which allows to discretize each microRNA separately into 3 patterns. MicroRNA can be either non-regulated, be up and down-regulated between the classes, or follow other microRNA regulation pattern. We filter out non-regulated and other pattern microRNAs from the data, as those are just not suitable for the application. This method allows also to score remaining microRNAs on how well they separate both classes. Not going into detail here, but we are using these robustness scores as additional optimality criteria in our algorithm. To find good distributed classifiers, we implemented an R Python framework that allows for data preprocessing and design and optimization of distributed classifiers. The baseline algorithm underlying the optimization process is a custom genetic algorithm that allows for multi-objective optimization, taking into account the previously mentioned robustness scores or other user-given microRNAs scores. Additionally, the workflow will soon allow to pre-optimize single circuits in distributed classifiers with logic solvers applied by us earlier to the design of single circuit classifiers. So this is work in progress, but for this we are using our previously mentioned logic programming based workflow to generate pre-optimized short functions that are later parts of the larger distributed classifiers and are an input to the workflow to speed up computation and increase the performance. However, as I mentioned, we're still developing this feature. So finally, I would like to show you some results on the distributed classifiers. We tested the classifiers first on the simulated datasets by introducing different level of noise by adding random outliers with certain probabilities. And we obtained above 89% accuracy, testing on novel unseen data for all those noise levels. Not really surprisingly, the accuracy is dropping with the increasing noise, and at the same time the complexity of classifiers is also rising. Then we tested our classifiers on real-world data, on liver cancer datasets and lung cancer datasets, and we noticed that these distributed classifiers perform rather better than single-circuit classifiers. Later, to increase the difficulty of the classification task, we introduced more diversity between the training and the testing datasets, by fully separating the normalization of those both datasets, and noticed that distributed classifiers seem to be much more robust to more heterogeneity between training data and novel unseen testing data, which is very important for the application. To sum it all up, multi-circuit classifiers and distributed classifiers perform very well on the simulated data and well on the real-world cancer datasets, outperforming single-circuit classifiers. I couldn't go into much detail here, but our classifiers also capture literature described as well as a bit more complex microRNA regulation patterns found in the data. What we think is important for the classifier design. First of all, proper data preprocessing and selection of good, robust microRNA candidates for our classifiers. Also, the classifiers should be trained to perform best on novel, unseen datasets. We think that the flexibility of the distributed classifier architecture seems to increase the robustness in case of very noisy and heterogeneous data. As an outlook, as mentioned before, we are working on the pre-optimization feature that relies on combining the two mentioned workflows together to increase the performance of classifiers and decrease the runtime of computation. At the end, I would like to acknowledge all the people and institutions mentioned in this slide, and I would be happy to take your questions.